Hi, this is Pradeep Haldar. Welcome to the yet another biology class relating to the cell. And today's topic is the cell wall and cell membrane. We know that every and each cell are covered by something. They are known as a cell covering. Now, when we discuss about the plant cell is bounded by a thick, non-living cell covering which is made by the cellulose, hemicellulose, lignin, pectin that known as a cell wall. Cell wall is absent in animal cell. In case of the animal cell is bounded by a thin living cell covering which is made by the protein and lipid that known as a cell membrane. So today's topic is cell wall and cell membrane. The first thing is Robert Hooke was the first scientist to observe the cell wall. Now we are going to discuss about the structure of cell wall. When we observe the cell wall under the general microscope or the light microscope, we can find that between the two cell wall one thinnest gap or layer present that known as a middle lamella. Middle lamella. So in the general structure of cell wall we can find the middle lamella. We know that during the cytokinesis the plasma store to form the cell plate which help to during the division of cytoplasm during the cell division. Now this cell plate are converted into the middle lamella and present between two nearby cells. Now the second one after the middle lamella we can find the primary cell wall. Primary cell wall means the cell wall which formed during after the cell division. So after the cell division newly formed cell wall are termed as a primary cell wall. And we know that different types of materials like cellulose, hemicellulose, pectin, lignin are stored in the cell wall by layer and layer. And the process by which helped by the storing process of cell wall or the materials is known as a assertion. So due to the assertion process, the cell wall starts storing of different types of materials and transform into or forms a new layer that known as a secondary cell wall. And the third one is maybe the tertiary cell wall. If the cell wall contains the xylem, then it is termed as a tertiary cell wall. Now we are going to discuss about the ultramicroscopic structure. Ultramicroscopic structure of cell wall. When the cell wall are studied under the electron microscope, we can find the different thing. The first one, some layer of the fluid substance present within the cell wall. They are termed as a matrix. And within the matrix, different type of fibrils are arranged. So, in case of the ultramicroscopic structure, cell wall contain two things. One is the matrix and another one is fibrils. Matrix means the fluid portion within which the fibrils are scattered or arranged, whereas the fibrils, thin separate fiber, are arranged either maybe the straight or parallelly, that known as a fibrils. These fibrils can be two types: macrofibrils. The thick fibrils arranged straight that are known as a macrofibrils, and the thin fibril which arrange parallelly, they are termed as a microfibrils. So, in case of the ultramicroscopic structure of cell wall, we can found that the thick fiber, suppose this is the thick fiber, these are the thick fiber arranged straight, and some thinnest fiber arranged parallelly, and within the portion the matrix present. Now these macrofibrils are formed by the almost 20 microfibrils. So 20 microfibrils are combined to form the macrofibrils, and each unit of the macrofibrils are termed as a micelle. Now between the different pores are present on the cell wall that known as a plasmodesmata. So what is plasmodesmata? Plasmodesmata means the pores present on cell wall that known as a plasmodesmata, which create connection between two neighboring cells or regulates the cytoplasmic movement between two cells. Now we are going to discuss about the function of cell wall. The first one, it's a general outer covering of plant cell. So it can fix the cell size and shape. It can protect the inner portion of the cell. In case of the plant, cell wall provides the mechanical support. And the last one, the plasmodesmata regulates the cytoplasmic movement between two cells. Now the next topic is cell membrane. Cell membrane, the outermost covering of the plant of the animal cell, and in case of the plant cell, it present innermost towards the cell wall. They are present thin living covering which is semi-permeable in nature and made by the protein and lipid that known as a cell membrane. Now Anton von Leeuwenhoek or A.V. Leeuwenhoek was the first scientist who observed the cell membrane for the first time. And the Pauli was the scientist who coined the term plasma membrane in the place of cell membrane. The next one is the component. Now cell membrane mainly formed by the three components. One is the lipids. Lipids means the fat molecules are combined to form the lipids. Different types of lipids may present within the cell membrane. It may be the phospholipid, maybe the sphingolipid, glycolipid, or the sterols. All are the present. And in case of the protein, according to the positions, the protein molecules of the cell membrane will be two types. It may be the intrinsic protein or the extrinsic protein. Intrinsic protein means the protein which attach with the cell membrane or present inside of the cell membrane, that known as the intrinsic protein. And the extrinsic protein, the protein which bearing distance with the cell membrane, that known as a extrinsic protein. Or on the basis of the function, protein can be different types. It may be the structural protein, it may be the carrier protein, or maybe some enzymes present. Now, the carbohydrate also present in the form of glycolipid and glycoprotein. These are the components which help to form the cell membrane. Now we are going to discuss about the structural model. The first one is sandwich model by Dawson and Daly. In case of the, according to the Dawson and Daly, cell wall is similar like the sandwich. In case the lipid molecules present between two protein layers. Now, two protein layers are arranged at the toppermost part, and between the two protein layers they are present the lipid layers, and suddenly they are arranged together like in that position. These are the protein layer and these are the lipid layer. Between the protein layer they are present the lipid layer. So it looks like the sandwich. That's why the Dapson and Derry provide the sandwich model. After that, the Robertson proposed the unique membrane model to discuss the structure of cell membrane. Unique membrane model. According to the Robertson. Unique membrane. Robertson thinks that cell membrane is a unique membrane. And when his models are observed by the electron microscope, suddenly he coined the term that cell membrane is a tri-layer membrane. The electron rays does not cause the protein layer. That's why form the dark band. Whereas the electron rays easily cause the lipid layer. That's why they form the light band. So, dark and light band continuously arranged in when we observe under the electron microscope. So, Robertson changed his term. But how? Robertson says that cell membrane is a tri-layer membrane, but these three layers are compact and behaving like a unique membrane. And the most scientific model relating to the cell membrane that known as a fluid mosaic model. Fluid mosaic model by Singer and Nicholson in the year of 1972-73. Singer and Nicholson. In case of the fluid mosaic model, first, cell membrane is a tri-layer membrane having the outer layer formed by the protein molecules having the thickness is 20 micron. These are the protein layer. And between the protein layer, the bilayers of lipid molecules present. Bilayers of lipid molecules present. These are the lipid, which are 35 angstrom thick. Now these are continuously arranged. These are continuously So, according to the fluid mosaic model, the lipid molecules provide the fluidity or liquid forms, and the protein molecules decorate at the both sides of the lipid molecules. Suddenly, it looks like the mosaic floor. That's why he coined the term fluid mosaic model. May some protein attach with the cell membrane. They are termed as a intrinsic protein or internal protein. And may some protein away from the cell membrane. They are termed as the external protein or extrinsic protein. The reason behind the intrinsic and extrinsic protein, it depends on the nature of lipid molecules. Generally, the lipid molecules having two parts. One is the head region and another is the tail region. If the head region having the hydrophobic in nature and the lipid also contain the hydrophobic in nature, then these two are repulsed and creating the external protein or extrinsic protein. If the protein molecules contain the opposite features relating to the head region of fat molecules, then these fat molecules are going to allow the protein to enter into the cell and creating the internal protein or intrinsic protein. Now we are going to discuss about the function of cell membrane. The first one is permeability. Permeability means providing permission. So through the cell membrane, different types of molecules or the gases and the solution are going to be entered within the cell. Now the first one is the impermeability. This type of membrane does not allow any substance except the gases to allow into the cell membrane. For an example, is the membrane of the polythenes or the parchment membrane. The semi-permeable membrane. Semi-permeable membrane means those are allowing the water to enter within the cell. They are termed as a semi-permeable membrane. And the third one is selective permeable membrane. Selective materials or the ions or the solution are going to be entered within the cell. That are known as a selective permeability. Human cell membrane is selective permeable in nature. The second function is transport. Transportation of materials take place through the cell membrane. Now transportation of materials may be take place by using of ATP or without using of ATP. If the transportation of materials occur against the concentration gradient, then the active transport take place. Active transport, where the energy is required, and the passive, if the transportation take place without using any energy, that are known as a passive transport. Even the osmosis and diffusion take place through the cell membrane. We will discuss about the osmosis and diffusion during the lab activity or some practical activity. And the third one is cytosis. The process by which cell receives something that known as a cytosis. Now endocytosis or the exocytosis. Endo means the inner. When the cell receives something or the something enter into the cell, that known as a endocytosis. It may be the food or the water. Exocytosis means cell discharging something. It may be the ejectory substance or the secretory materials. Now endocytosis can be two types. It may be the phagocytosis. It's known as a cell eating, the process by which the food materials enter into the cell, that known as a phagocytosis, or the process by which water enter into the cell, that known as a pinocytosis. I think that's enough for today. Tomorrow we will discuss about the protoplasm, and if you have any doubt, you can mention it into the comment box.